Hello. Today I'll talk about what happens when the hormone reaches its target. As I've noted before, including in an email as well as your main source of study, the handout I gave you, it is the chemistry of the hormone that is defining what kind of response the target cell can have. So, large hormones, like large proteins like growth hormone, prolactin, cannot enter the cell through the membrane. Smaller molecules that are polar, uh, like for instance amines, those are derivatives of amino acids that are polar, they cannot enter the cell because they cannot pass through cell membrane because polar molecules are hydrophilic and the fat that makes up the cell membrane is hydrophobic. As you know, the oil and water don't mix. That's how you can get lava lamps because you have oil going through the water, and and thus polar molecules or very large molecules cannot pass through the cell membrane, which means they have to act via membrane receptors. Membrane receptors uh, are proteins that are embedded in the membrane, and at least part of it is on the surface of the membrane and has a 3D configuration that can fit the shape of the hormone like a lock and key. There are several different types of membrane receptors. Uh, for instance, a membrane receptor can be a relatively simple protein that acts as an enzyme itself. So when a hormone binds from the outside, this changes the 3D shape of the receptor, and the inside end of that protein receptor acts as an enzyme, which means it catalyzes some kind of a biochemical reaction inside of the cell, thus changing temporarily the properties of that cell, what that cell does. Um, that's relatively rare. Another type of membrane receptor is an ion channel. So when a hormone binds on the outside, lock and key, that changes the 3D shape of the protein that is spanning the thickness of the membrane. And that change of the 3D shape, let's say untwisting it, opens a channel, opens the hole through the entire length of that protein. So now ions can go in or out of the cell, changing the properties of the membrane, the voltage of the membrane, which is changing the property of that cell, which means that cell can do something differently than it did a second ago. The most common uh, receptor that is on the membrane is the 7 transmembrane G protein receptor. Those were actually discovered here at Duke and um, received the Nobel Prize for that. 7 transmembrane G protein associated receptors are long chains of proteins that transverse between the inside and outside of the membrane 7 times. They go in and out, in and out, in and out 7 times. So there's going to be one end, which is outside, which is capable of capturing the signaling molecule, in this case, the hormone signaling molecules of other types use these too. For instance, smells, deodorants are going to attach themselves to these kinds of receptors. Uh, Receptors for neurotransmitters in the nervous system are this type of receptor. But now we're talking about hormones, so let's say that a hormone has attached to that first tip, the beginning of the hormone of the receptor. The receptor then transverses the the membrane 7 times, and the last end, the opposite end of the protein, is on the inside of the cell. That protein is associated with the G protein. The G protein has three subunits, alpha, beta, gamma, and this is all tied together. What happens when a hormone attaches itself to the 7 transmembrane protein receptor? It takes a little energy to dissociate itself the from the G protein. This is Freddy, who has been bothering me since we started. Um, the 7 transmembrane protein takes that energy by cleaving off a phosphate off of GTP, guanosine triphosphate, leaving GDP, that diphosphate, hanging. So the energy released when that bond, the phosphate bond, is broken, is sufficient to remove the alpha subunit of the G protein away from this complex. That alpha subunit then attaches itself to adenylyl cyclase, which is an enzyme also embedded in the membrane, uh, which is close by. Remember the whole fluid mosaic membrane model, that it's a bunch of fat molecules and then there are protein molecules kind of floating around. They can move pretty easily through that sea of fat molecules, and so the alpha unit, a subunit of G protein, once receiving energy, can move away and attach itself to the adenylyl cyclase. This complex now energizes what? The second messenger. The hormone itself is often called the and the first messenger. The second messenger is what is on the inside of the membrane, and that is cyclic AMP. Cyclic AMP gets energy, adds a phosphate, and which energizes some kind of uh, an enzyme, which is going to energize another enzyme, which is going to energize another enzyme, another one, until something is happening in the cell. What is interesting about this is when the one molecule of the hormone attaches from the outside, the cyclic AMP gets turned, many molecules of cyclic AMP get energized, and each one of them is going to energize multiple copies of that first protein enzyme, and each one of those enzymes is going to energize multiple molecules of the second enzyme, and each one of those is going to uh, energize uh, multiple copies of the third enzyme, which means that the single molecule of the hormone has resulted in thousands and thousands and thousands of energized last enzyme, which means it's a very, very big and very fast effect. That cell is now doing something completely different and a large scale, at a really large scale. There are thousands and thousands of molecules doing some kind of a biochemical reaction that they didn't a millisecond ago. There's an enzyme that removes all of this. So this is a very uh, short effect, and that kind of forces the hormone to detach. So the body is using a lot of hormones in this way because a lot of functions have to be very precisely timed. So the onset of the response is immediate and really, really fast. The result is huge and it stopped immediately, very fast. So you tell the cell, do this really big and now stop. Uh, a lot and lot of functions in the body have to work that way, which is why most of the hormones are either proteins or peptides or polar molecules. Polar meaning amino acids that have been modified in some way, and they have polarity, either positive or negative charges somewhere on them. Not every function requires this. Some functions uh, require inhibition and can still work through the 7 transmembrane G protein uh, uh, associated receptor, sometimes called the serpentine receptor because it looks like a snake, like a serpent. And one example is somatostatin, which is the factor or hormone that is secreted from the hypothalamus, goes into the anterior pituitary to inhibit growth hormone. It inhibits the growth hormone by uh, by stimulating 7 transmembrane receptor in such a way that has the opposite effect, inactivating the G protein, stopping the activity of adenylate cyclase. So sometimes it can be inhibitory. It's rare, but it happens. Sometimes it works that way. But there are other functions in the body that don't need to be that big, or don't need to be that fast, or should not last that short. For those kinds of functions, the body uses different kinds of molecules. Those are going to be nonpolar molecules. Those are going to be hydrophobic molecules, which means they can go through fat. Fat can go through fat, like water cannot. Those molecules are going to enter the cell. They're going to pass through the membrane. They're going to enter the cell and float around the cytoplasm. Now, remember, the cytoplasm is not just gunk and goo, it also has cytoskeleton, various uh, micro, various types of uh, protein threads and whatnot, and the hormone is probably going to attach to one of those threads and go down into the nucleus or wherever, and relatively easily find and bind to its receptor, which is found either in the cytoplasm and in the nucleus. Now, remember, nuclear membrane has pores, so those receptors are likely to go back and forth. They're going to be in the cytoplasm and then move in the nucleus back into the cytoplasm. So that nuclear or cytoplasmatic receptor can be found in each one of those two compartments at any given time. Uh, so, what kind of molecules are those? Those are steroids. Those are all derivatives of cholesterol, and we use quite a lot of them. Various estrogens. When I say estrogen, it's not one molecule, it's a whole class of molecules like estradiol, estrione, estrol. Then there are androgens like testosterone, several others, progesterone. Then there is cortisol, aldosterone. Those are all derivatives of cholesterol. And then there are nonpolar molecules that are that can enter the cells because they're small and nonpolar, although they're not themselves fatty. Those are derivatives of amino acids that don't have any uh, positive or negative charges on them. Now, your book says that the only one of those is thyroid hormone. That's not true. Melatonin is also a nonpolar derivative of amino acid. Instead of thyroxine, it is tyrosine. It's a derivative of tryptophan. So those two molecules, melatonin and thyroid hormones, either 3 or 4 iodothyronine or thyroxine. The 4 iodothyronine is also called thyroxine. So those two thyroid hormones and melatonin also enter easily through the membrane into the cytoplasm. So how do these guys work? They work by attaching themselves in onto the receptors that are floating around the cytoplasm or the nucleus. The exception of thyroid hormones, which are already whose receptors are already attached to the DNA. So steroid receptors, melatonin receptors, they're going to catch the melatonin or a steroid, and then they're going to together go into the nucleus if they're not already there, and that complex, the hormone receptor complex, can recognize a particular sequence of DNA. Now, that particular sequence of DNA are promoters. Those are promoters for particular genes. And it's never going to be just one gene. There's going to be several genes in several different places in the genome, on several different chromosomes, that are going to have genes that have exactly that same promoter, the same sequence of the promoter. So let's say an estrogen binds its estrogen receptor. That complex, which now has a different 3D shape, can recognize a particular estrogen binding promoter site, which is then going to turn several genes in the genome on. And now you have that DNA being transcribed into RNA, RNA edited, and moved out to the nucleus onto the endoplasmatic reticulum, binds on ribosomes, and translates into protein. Now the cell has a protein that it didn't have before. And this process is much slower. It took me longer to explain how it goes, right? All that, moving around, that's, finding each other is slower. Migrating into the nucleus is slower. Finding those binding spots, sometimes you have to move some histones around in order to bind. Then turning all those genes have to be transcribed and then edited and then moved out to the cytoplasm, binding to ribosomes, translated, then post-translational modification of proteins. This lasts a long time, at least minutes, usually hours, days, even longer. But the trick is that this lasts a long time. The effect doesn't really have kind of a stop switch, an off switch. It is going to stop once, let's say, everything is degraded, which can happen actively or passively by the cell, or it doesn't have to happen at all. So, for instance, the effect of melatonin is going to degrade when the organism sees the light. Then it's going to get a switch to degrade this complex. For many of those hormones, like steroid hormones, estrogen, testosterone, progesterone, there is no off switch. So once they alter the properties of the cell, now the cell is making different kinds of protein and doing new things. There's no going back. That cell is now completely altered. Usually, this happens early in the embryo, and in the last video, I explained the example of testosterone priming or organizing the cell, and then subsequent, much later, years later, exposure to the same hormone or a different hormone is going to have an activating effect. This cell is now different, and now it's capable of responding to this hormone again with having by having some kind of other response, some kind of other reaction. So that is how nonpolar hormones work. Slow, long lasting. There are functions for which you need that. As I said, thyroid hormone receptor is already attached to all the necessary uh, promoters of the genes that thyroid hormone is supposed to turn on, and so the thyroid hormones transports itself down some kind of microtubules or something like that into the nucleus and binds to the thyroid receptor that's already bound onto DNA. Once the hormone and receptor bind to each other, that changes their 3D level and energy, which starts the process of actually transcribing the gene right next to it. So this is how most of the hormones, most of the signaling molecules work. There are some molecules that act in autocrine and or paracrine fashion. Uh, those are usually very, very small molecules or gases. Uh, One of those is nitric oxide. Nitric oxide, you need an enzyme that activates and builds nitric oxide, and nitric oxide is going to uh, act on the neighboring tissues. It's going to diffuse because it's really, really tiny. It's going to diffuse through the cells and uh, energize them in some way. The most common effect of nitric oxide is vasodilation, opening of the capillaries, opening of the blood vessels, which is why it's the basis of Viagra. It opens up the blood vessels in the genitals. However, you have probably heard that the Viagra was discovered by accident. The study was really for a vasodilator for systemic for people having asthma and those kinds of things where or uh, other problems where you might have vasoconstriction for some reason and you want to open up those capillaries. I'm assuming, as kind of health professionals, that you love Grey's Anatomy. There was an episode in Grey's Anatomy where Meredith Grey and Griggs were on an airplane. An airplane had a really, really horrible turbulence, and so they had to deal with, like, 3 or 4 or 5 different patients, doing even surgeries on some of them. And one of them was kind of an obnoxious guy that was trying to get attention on himself, and suddenly he collapsed. And Griggs diagnosed that he has pulmonary hypertension, which means his uh, his blood pressure in the lungs was so high that blood was starting to leak into the lungs. He couldn't breathe. So one thing to do is to open up the diameter of the blood vessels in the lungs, so all that blood can actually go through. Reduces the blood pressure in there. What can you do? Well, he asked the cabin, like, is there anybody here who has a vial of Viagra? And there was this like old grandma saying, "This is my husband's," and he ground some Viagra or whatever and injected into the patient, who then survived. He was fine. So nitric oxide is a vasodilator, and it is, as a gas, has a very small molecule. It can easily pass through various membranes, and it acts mostly in a paracrine manner. All right, thank you.